So hi everyone, I'm Wang from Mainland, Germany. Yeah, I'm currently a postdoc at Key University. So I'm the colleague of the previous president, Dustin Hunt. So my title today is about the evolution of antibiotic resistance island in plasmid genome evolution. Lateral gene transfer play a very important role in the bacterial genome evolution. And I focus here the evolution of mobile genetic elements and antibiotic resistance genes, specifically in plasmids. And here, before I introduce my work, this is a theoretical framework. There are three components: antibiotic resistant gene, ARG, transponses or recombinases, I call it SSR, and the third one is plasmid. So the first two components, ARG and SSR, they form combinations, and the transponses would translocated ARGs, and furthermore, the combinations will interact with plasmids. And during the evolution, if there's a selective pressure, the combinations will accumulate in plasmids and form specific patterns. Yeah, and I call it resistance island. And the successful plasmid will distribute the previous two basic components, antibiotic resistant gene and transponses, furthermore to the gene pool. And in this way, it forms an evolutionary cycle. And this cycle is the process from pieces to islands. So my main objective of this study is to study the contribution of mobile genetic elements, mainly transponsible elements, in the evolution of plasmid resistance. Yeah, and examine the interaction between the transponsible elements and plasmids. This is my basic workflow. I focus on three genera: Asia, Salmonella, and Klebsiella. They are famous for multi-drug resistance plasmids. There are multiple plasmids and they carry multiple antibiotic resistant genes. I build gene families. I identify the ARG and the SSRs, and I search in the plasmid genomes the conserved collinear blocks, the conserved genetic neighborhoods, which contain pairwise of significantly co-occurring ARGs. In this way, I first find out my potential objects and align those conserved collinear blocks to transponsible elements. And I perform a network analysis to analyze the process of resistance islands assembly. So the first result is, in my data, 84% of the ARGs in multi-drug resistance plasmids are located in those islands in plasmid genomes. So this is a network, and each of the nodes is a gene family, either ARG resistant gene or recombinases. Both of the type of gene, they are super conserved in my data set. So I use the isolation data of the bacterial genomes, and I analyze the growth of this network along the timeline. I found the resistance network, the islands, assemble quite fast. And I observed that there's an early saturation in the year about 2014. Even though the sampling density of plasmid, the sequencing plasmid, increased significantly after the year of 2014. And I show that I embedded this knowledge into the plasmid lineages. Here, PTU, plasmid taxonomy units. Here, this plot, I show that the ARG islands are restricted to specific PTUs. Mainly in the plasmid with size larger than 20 kilobase pair, but not only the large plasmid, they carry ARG islands. Furthermore, this is an example of the details of three groups of island pieces. I show here the island pieces. They are specific to different PTUs. For example, the green, the light blue, and the dark blue. These three groups of island pieces, they they distributed unevenly in three groups of PTUs. To summarize, first, I found the majority of plasmid resistant genes can be found in those islands in plasmid genomes. The island assembly is a rapid process, and we observed an early saturation in this process. And I show here the ARG island is PTU specific. So specific plasmid lineages carry ARG island, and the ARG island pieces are PTU specific. So which means there are barriers to island pieces transfer. And the barriers could be related to plasmid genetics, the host range, and we also need to consider the plasmid evolutionary history. Yeah, thanks to my supervisor, and you're welcome to read this paper. If you are interested in lateral gene transfer, mobile genetic elements, or mobility of antibiotic resistant gene, you are more than welcome to come to say hi and talk. Yeah, thank you very much.